Good afternoon, my name is David and I am a PhD student at the University of El Paso, and today I'm going to present my poster titled Behavioral Analysis of the Zebrafish with Mutations of the HFC1A gene. The gene we are interested in is known as the HFC1 gene. This gene is a transcriptional cofactor that regulates the expression of over 5,000 different downstream target genes. And mutations in this gene is known to cause CVLX syndrome in humans. CVLX syndrome is mainly characterized by metabolic disorders and intractable epilepsy. Previous studies from our lab have demonstrated that mutation of HFC1A in zebrafish increases neural precursor cell numbers and is associated with hypomotility. However, these results were obtained using haploinsufficiency model, which does not mimic the missense mutation in CVLX syndrome. Therefore, our goal is to create a homozygous viable mutation that will mimic more the CVLX syndrome. For this reason, we developed a missense mutation in the HFC1A gene that we call Co64, which is homozygous viable, allowing us to study the behavior in homozygous carriers. We hypothesized that a missense mutation of HFC1A increases neural precursor cell and causes abnormal locomotion behavior. Therefore, our aim is to identify the role of HFC1A in locomotion behavior and brain development. To better understand how mutation affect neuronal networks and patients' behaviors, we can use the zebrafish model to understand neurodevelopmental disorders. To accomplish our aim, we use zebrafish larvae to analyze the gene expression at 5 days post-fertilization using the qPCR. Then we use the ZebrafishBox technology, which allowed us to track the swim patterns of larvae at 5 days post-fertilization. And finally, we use the flow cytometry to analyze neural precursor cell counts in homozygous carriers to correlate deficits in brain development. We first validated our homozygous carriers by using 2 sets of primers. One set will be specific for the amplification of wild-type sibling, as shown in Figure 1a. The second set of primers will be specific for the Co64 allele, as shown in Figure 1b. We then analyzed the expression of homozygous carriers and we found an 80% decrease in gene expression, as shown in Figure 2, which validated our unique missense mutation is being affected at the RNA level. When we found that the expression levels of our Core64 model was significantly decreased, we asked if that will have an effect in brain development. Based on previous study from Castro et al. In our lab, we hypothesized that neural precursor cells would be abnormal. To answer this question, we analyzed neural precursor cells using a SOX2 transgene line as a marker. We used the flow cytometry technique to detect abnormal SOX2 GFP levels, and we detect a 10% increase in our Co64 mutants when comparing it with the wild-type sibling as shown in Figure 3b and 3c, indicating defects in brain development. In our previous studies by Castro et al., we found hypomotility associated with neural precursor cells. Therefore, to investigate the impact in our homozygous viable Co64 allele, we perform behavioral analysis. In Figure 4, we found that upon light stimulus, our homozygous mutant larvae exhibited a decrease in motility when comparing it with the wild-type sibling, indicating that a mutation in the HFC1A affect locomotion behavior. Collectively, our data suggests that the Co64 allele, which is homozygous viable, parallels the neural precursor cell and behavior phenotypes present in previous alleles. To conclude, I would like to acknowledge my mentor, Dr. Quintana, my lab mates, and the funding from NIH, as well as the SDB for the opportunity of participating. Thank you.